The next part of Eubacteria characters is reproduction. Bacteria participates in 3 types of reproductions. Those are vegetative, asexual and the sexual method of reproductions. First, let us see which type of vegetative reproduction is exhibited by bacteria. Here, this is the reproduction which occurs during favorable conditions. And as it is taking place during favorable conditions, it is considered to be the most common method of reproduction that occurs in the bacteria. And this vegetative reproduction occurs by a process called as binary fission. So, binary fission is a common method of reproduction which occurs in bacteria. This binary fission is the process where the bacteria will divide into 2 daughter bacteria by cell division. And here the time taken for one binary fission is approximately 20 minutes. Or nearly it will take 20 minutes, such that in every 20 minutes the number of bacteria will become double. And generally here, as I said, that the division of the cell will take place, and the division will start in the region where the mesosomes are present. The place where the mesosomes are there, from there the point of binary fission is present. And here first thing is the DNA, circular naked DNA which is present, will replicate and slightly elongated. Slightly get elongated, that is considered this as a bacterial cell with a cell wall and the plasma membrane around it. First, the nucleus will gradually elongate like this along with the replication, and it will be converted into 2 daughter nuclei. And at the same time, a small constriction will start from the peripheral side of the plasma membrane or the cytoplasm. So, this is a site where the mesosome is present, and this little constriction that is present will gradually move towards the center, and this constriction is also moving towards the central. That is, the constriction will start in the peripheral side and move towards the central. So, the growth is called as a centripetal growth of the constriction. Once this constriction will move towards the center, both these constrictions will join at one place, and gradually in the next step, the cytoplasm is divided into 2 parts with a nuclei inside them, and in the same way the cell wall will also start a constriction. So, this constriction will start at the same place where the constriction of cytoplasm have been started, and the constriction of the cell wall will also move towards the center in the centripetal manner and divide the bacteria into 2 daughter bacterial cells. So, likewise the binary fission will occur by a process called as division in a centripetal manner, leading to the formation of 2 daughter bacteria. And here this bacteria, once 2 daughter bacteria have been formed, the survival of the parent bacteria will not exist. But it is not the death of the bacterial cell, but it is the parent cell itself is divided to form into 2 daughter bacteria. Likewise, for every 20 minutes, the number will become double to that of the actual existing number. So, this is a common method of reproduction which occurs in bacteria, that is vegetative reproduction by binary fission. The next method of reproduction is the asexual reproduction. Asexual reproduction occurs during unfavorable conditions. As it is taking place during unfavorable conditions, so this occurs rarely in its life cycle, and here this asexual reproduction will occur by formation of special structures called as endospores. So, when unfavorable conditions are there, the water quantity is less inside the cytoplasm. At the same time, the cytoplasm contains an special type of chemical called as dipicolinic acid. So these are two characters which are exhibited by the cytoplasm, where the water quantity will decreases and contains dipicolinic acid. And here the bacterial cell will form an additional layer, a thick layer around it. The thick layer, a thick wall is formed. So this thick wall is made up of 4 layers. Those 4 layers are, outermost one is called as exosporium, which is made up of lipoproteins, that is lipoproteinaceous. The second one is called as spore coat. This is made up of a special protein called as keratin. And the third layer is called as cortex. Cortex is made up of peptidoglycans. And the fourth one is called as spore wall. This spore wall is made up of proteins. So like this, the thick wall is formed by 4 types of layers, which appears to be very thick in its nature, and it will sustain during unfavorable conditions because of this thick wall, and this thick wall is resistant to high temperatures. And once the favorable conditions are retained, the thick wall will gradually dissolve, and the cytoplasmic contents will produce a new layer around it, and it will be surviving or forming into a new individual. Likewise, during unfavorable conditions, the thick wall will protect it from the high temperature and abnormal atmospheric conditions. Likewise, the endospores are helping them to sustain during the unfavorable conditions. The third type of reproduction is the sexual reproduction. Sexual reproduction means the two parents have to participate, and gamete formation and meiosis should be there. But here in the sexual reproduction of bacteria, no gamete formation. At the same time, no meiosis. When no gamete formation, no meiosis, then we should not consider it as a true sexual reproduction. But why we are considering that bacteria have sexual reproduction means because here the genetic material is transferred from one bacteria to other bacteria. So because of only this character, we consider it as a sexual reproductive process. So here this is not a true sexual reproduction, and here the transfer of genetic material will occur by 3 methods. Those are 1. Conjugation, 2. Transformation, 3. Transduction. So these are the 3 different methods of transfer of genetic material from one bacteria to the other bacteria. Let us start with the first one, that is conjugation process. So here in conjugation, the 2 bacteria will participate in the process. Here the bacteria which contains F factor, that is the fertility factor will be there. So the one which contains this fertility factor in the form of plasmid, so we call that as a F plus bacteria. And the one which lack F factor, that is the bacteria which lack the fertility factor, we call that as a F minus bacteria. Always the conjugation will occur between a F plus bacteria and a F minus bacteria, and during this process what happens is the plasmid, the F factor which is present in the F plus bacteria, will undergo replication, and it will form one more extra copy of the F factor. Thus extra copy will be transferred from the F plus bacteria to the F minus bacteria. That means once the conjugation have been completed, the F minus will also be converted into F plus bacteria because it contains fertility factor which have been transferred from the F plus bacteria. And here, apart from this one, HFR bacteria are also present. So HFR bacteria means high frequency bacteria. What is the difference between HFR and F plus bacteria means one similarity is there, that is both of them have the plasmid, that is fertility factor is present. But in F plus bacteria, the fertility factor is present in the cytoplasm and not integrated with the nucleoid chromosome. Whereas in HFR bacteria, the plasmid part or the F factor is integrated with the nucleoid DNA. So that is the difference between the HFR bacteria and the F plus bacteria. The conjugation may also occur in HFR bacteria. So likewise, when the conjugation is taking place, the F factor will be transferred from one bacteria to the other bacteria. So here, this process of conjugation was first discovered by Lederberg and Tatum in Escherichia coli bacteria. So this is the first method of genetic material transfer that occurs in bacteria. And here in the process of conjugation, as we have discussed in the morphology of bacteria, that is they have the sex pili. The sex pili will contains a hollow core through which the genetic material will be transferred from one bacteria to the other bacteria. That is the sex pili will act as a conjugation tube during the process of conjugation. The second method of sexual reproduction is transformation. Here in the process of transformation, there is no agent or vector which help in the transfer of the genetic material from one bacteria to other bacteria. Here the naked DNA is taken directly from a environment by the recipient bacteria. So here what happens is, once in the environment when a bacteria undergoes lysis, so we can say and donor bacteria which undergoes lysis, and when it undergoes lysis, it will release the DNA into its outer environment, and this DNA or the genetic matter will undergo small fragments, and the recipient bacteria which is present in that same environment, it will start absorb this DNA or take up the DNA which is present in its surrounding environment. That is naked DNA fragments are received by the recipient bacteria, and once this bacteria have been received by the recipient bacteria, that will get integrated with the DNA of the nucleoid of the recipient bacteria. That is the way the integration will be taking place. So here the absorption of the bacteria directly from the environment is called as a transformation process, and here this process was discovered by Griffith scientist in Streptococcus pneumonia or pneumoniae bacteria. So it was first discovered by Griffith scientist in Streptococcus pneumoniae bacteria. So this is a way how the direct absorption of naked DNA from its environment without any vector or the agent, this process of absorption of DNA is called as a transformation process. In the sexual reproduction, the third method is a transduction. Transduction process of sexual reproduction was first discovered by Lederberg and Zinder scientists in a bacterium called as a Eubacteria. Salmonella typhimurium. And here in this process the bacteriophages, phages means the virus particles, are helping in the transfer of genetical material from one bacteria to the other bacteria. The viruses which attack the bacteria are called as bacteriophages. These bacteriophages will help in the transfer of genetical material from one bacteria to the other bacteria. So here actually the bacterial cell wall is resistant to enzymes, but here the bacteriophages will have an enzyme called as a lysozyme enzyme. So this is the enzyme which can dissolve the bacterial cell wall. So this enzyme is secreted by the bacteriophages or the viruses which infect the bacteria. So because of that lysosome, the bacterial cell will be dissolved and it will help in the transfer of genetical material from one bacteria to the other bacterial cell. So likewise in all these processes of sexual reproduction, nowhere we have seen the gamete formation nor the meiotic process, but only process is that the transfer of genetical material from one bacteria to the other bacteria. That's why here we should not consider it as a true sexual reproductive process. After reproduction, the next one is the economic importance of bacteria. So bacteria have economic use. So that is some of the bacteria are useful to the humankind, and at the same time some bacteria are harmful to the humankind and to the other animals and plants also. So based on that one, let us see the useful and also the harmful characteristic features related to the bacteria. Let us start first with the useful activities. There are many such bacteria which are useful to the humankind and to the plants and animals. So let us take some of the examples, like first in the agriculture field. In the agriculture field, these are there are some bacteria which are considered to be the nitrogen fixers, or that is they are useful for the nitrogen fixation. So already we had discussed earlier that rhizobium is one such bacteria which will help in the nitrogen fixation in the leguminous plants. So likewise rhizospirillum, like some other examples are there which are acting as a nitrogen fixers, that is rhizobium, Azotobacter, Rhodospirillum. Such type of bacteria are useful for the plants in the form of nitrogen fixation. And the same time some bacteria are there which are considered to be the ammonifying bacteria, that is called as they are useful for the process called as ammonification. Ammonification is a process where the complex nitrogenous organic nitrogenous compounds are converted into ammonia. Organic nitrogenous compounds are converted into ammonia. So this is done by some species of bacteria, that is like in bacillus. And at the same time some of them are useful for nitrification. Nitrification is a process where ammonia is first converted into nitrite and nitrite is converted into nitrate. So this is a process where nitrogen is made available to the plants. At the same time some of them are used as biopesticides. As we know, nowadays in the agriculture, crops are being affected by the pesticides, but chemical pesticides are very much available. Here the chemical pesticides are having some damage or harmful effect on the crop and also on the field by causing some pollution. In place of that one, nowadays biopesticides are very much useful, and these are economically useful and also non-polluting. We can call them as a eco-friendly in their nature. The best example for this biopesticides is Bacillus thuringiensis. In short form we call it as a BT. So here the scientists had developed a technique where the Bt genes are transferred into the plant species, which can help in the success rate of crop production. That is the way how this bacteria are useful in the agriculture field. The bacteria are useful in the industries also. Like first one, curd preparation. So in our houses, naturally we every day the curd is prepared by adding little amount of curd to the lukewarm milk. So in that process, the curd which have been added to the lukewarm milk contains some millions of bacteria. That bacteria is called as lactobacillus. These lactobacillus have the ability to coagulate the casein protein which is present in the milk. In that process of coagulation, the milk is converted into curd. That is the way how this lactobacillus is used in the curd preparation. The next one is curing of leaves of tea and tobacco. So in the process of curing, the leaves are made into softened such that the economic significance of that leaves can be exploited. So that is the curing of leaves of tobacco. Bacillus megatherium is used. So as we know that the tobacco leaves are used in preparation of the tobacco products like cigar and cigarettes, etc. So tea used as an alcoholic compound or beverages. Okay. Then retting of fiber. So here the fiber retting, that is separation of the fiber, can be done by using some bacteria, that is Clostridium species. Some Clostridium species are used for this purpose. And apart from this one, there are some medicinal values are also there. So large scale production of some medicines is done by using some bacteria in industries. So that is Escherichia coli, which is used in the preparation of insulin hormone. As we know that naturally the insulin hormone is produced in the human body to control the sugar levels in the blood. But as the age increases, the production of the insulin hormone will decreases. And in order to compensate that, the scientists had used E. coli to produce insulin hormone. So that insulin hormone which is produced by using E. coli is called as humulin. Likewise, this is one other medicinal value. And apart from this one, there are many such bacteria are there which are used in the production of antibiotics. Production of bacteria in the antibiotics like streptomycin is produced from Streptomyces griseus. The same way, oxytetracycline, which is produced from Streptomyces zymosus. So likewise, neomycin, like many such type of antibiotics are produced by using some bacterial species. So apart from this one, tetracycline, bacitracin, or some of the other examples of antibiotics which are produced in the industries by using the bacteria, that is they are exploiting the useful characters which are present in the bacteria for the well-being of the human beings, animals and the plants. Okay, nowadays the new techniques have been coming into existence, that is genetic engineering. In the process of genetic engineering, the scientists are using the plasmids which are present in the bacteria. The most useful natural plasmid in the genetic engineering is the Ti plasmid, which is present in Agrobacterium tumefaciens. Once this the tumor inducing genes have been disarmed, the Ti plasmid can be used in the transfer of genetical materials or transfer of desirable genes from one organism to the other organism. And scientists had developed the artificial plasmids also based on the structure of the natural plasmid. The best known artificial plasmids are PBr322. So such type of plasmids have been nowadays used in the genetical transfer or the transfer of desirable genes from one bacteria to the other and from one organism to the other organism. That is the way how these plasmid characters which are present in bacteria are exploited in the genetic engineering in the industry level. In the economic importance, the next one is harmful activities. At the same time, when you are talking about the useful activities, there are many such bacteria which are causing harm to the humankind, plants and also to the animals. So let us take some of the examples there. Here one is spoilage of food. So there is one bacterium called as Clostridium botulinum. This is a bacteria which is causing spoilage of food, and how it is doing the spoilage of food means by releasing a toxic material. One toxin is released, the toxin material is called as botulinum. So this botulinum will cause spoilage of food. That process is called as botulism. Sometimes this whenever the botulism has been caused, it may lead to the death of the organisms also. Apart from this one, there are many such bacteria which are causing diseases to human beings, animals and also to the plants, like leprosy, which is caused by Mycobacterium leprae, and tuberculosis, which is caused by Mycobacterium tuberculosis, diphtheria, which is caused by Corynebacterium diphtheriae. So likewise anthrax, Bacillus anthrax, likewise many such diseases are caused by the bacterium in the human beings. At the same time, there are many such diseases which are being caused by bacterium in animals also, like tuberculosis, diarrhea in poultry. Many such examples are there which are causing much damage to the production of the livestock, that is the animals. And in plants also, many such diseases are caused by bacteria, like citrus canker. Citrus canker is caused by Xanthomonas citri. This is one disease. And fire blight of apple. Many such diseases are caused by bacterium in the various types of plant crops also, which are causing much damage to the agriculture field and to the farmers. So this is about the various economic importance related to some of the bacterial species.